So let's get started. Today we have our first speaker of the day, Dr. Jess Bills from the Department of Biology, College of Arts and Sciences. So please join me in welcoming Dr. Jess Bills.
Okay, so I think we can all appreciate how much our senses impact our perception of the world around us. And vision is one of the most impactful senses in that perception. And the loss of vision can be quite devastating. Retinal degenerative diseases affect millions of people worldwide, and such diseases like age-related macular degeneration and glaucoma will eventually lead to vision loss. Now, these current treatments for these diseases are quite promising. However, the issue lies that they're not diagnosed until symptoms occur, and oftentimes symptoms don't occur until those neurons start to degenerate. And once we have enough damage in those neurons, we will start to lose vision. And obviously, as humans, unfortunately, we do not have the ability to regenerate our neurons. But what if we did have that ability? Because we do have animals that can regenerate their retinal neurons, but so far we have not seen a mammal that has this ability. But if we could find such a mammal, it could really provide us so much insight onto the questions of how and why retinal regeneration occurs. So let me tell you about this little rodent. The spiny mouse, genus Acamus, is found in parts of Africa and the Middle East, and they do have remarkable regenerative ability. In recent years, it has been shown that they have functional regeneration in their spinal cord, kidney, and heart, and complete scarless regeneration in their ear pinna, skin, and skeletal muscle. So if you can imagine, this is a really exciting animal, and it really lends way to the question of what happens to their retinas after retinal damage. So the retina is a very thin layer of tissue in the back of the eye, and it is responsible for converting photons of light into an electrical signal that the brain can read, and it sends that signal to the brain via the optic nerve. The retina is very important, obviously, and all of the neurons are important. We have 5 different types of neuron and 1 intrinsic glial cell, but for the sake of time, I'm going to point your attention to 2 cells that I'm going to show you a lot of today. That first cell is the retinal ganglion cell, and their axons culminate in forming that optic nerve, so that sole pathway of that visual process to the brain. And then the next cell I want to tell you about is that intrinsic glial cell, the Mueller glia. They have lots of important responsibilities, from just maintaining general homeostasis and a healthy retina to actually dictating whether a species can undergo retinal regeneration. For instance, in permissive species like the zebrafish that can regenerate their retinas, their Mueller glia is the primary cell that drives that regeneration, and it responds to the regeneration in a pro-regenerative manner. It responds to the injury in a regenerative manner. For instance, after retinal injury in the zebrafish, the Mueller glia activate, and by doing so, they proliferate, meaning they divide and produce daughter cells. And those daughter cells here actually have the cellular profile of a retinal progenitor. So they're a very young neuron, and they are capable of maturing into any of the 5 different types of neurons that are needed to replace after injury. So we see this in the zebrafish. But in mammals so far looked at, including laboratory mouse, this does not happen. The Mueller glia does not respond in this way. However, we have not yet had the opportunity to investigate an animal with such regenerative ability. So what happens when we damage their retinas? So in order to answer this question, we adapted a well-documented protocol using a toxin NMDA and injecting it to ablate or kill the ganglion cells. This is a well-used protocol used in glaucoma research, as glaucoma affects those ganglion cells. So again, we use that toxin that's going to ablate those ganglion cells. So the first thing we want to look for is that the damage is being done similarly to what we have seen in other mammals and the spiny mice. And what you can see, so I'm going to orient you to this data real quick. You'll see a lot of really beautiful images of retinal cells throughout this talk. And this first row here is our positive injury control. So we use the mus as a positive control. their response to this injury is very well documented. So up top, we can see the mus musculus, or the laboratory mouse, as our control, and the bottom is our spiny mice, or ACMIS. All of the quantification graphs you will see have the same 4 conditions for each time point, the first 2 being our control mus, control in treated conditions, and then the second being our spiny mice, or ACMIS, control in treated conditions. So going back to the ganglion cells here in the healthy retinas for that PBS injected, we can see those ganglion cells lit up really nicely with that green stain. So this bottom row right here is all of the ganglion cells. And I hope you can appreciate over the first 3 days after injury, highlighted in the red square, you can see a reduction of those green cells. So we are losing those ganglion cells. So the damage is being done appropriately. Additionally, you will see gene expression graphs shown as dot plots throughout this data, where the deeper color pink, the higher gene expression of those cells. For instance, here we have 4 genes regularly associated with mature retinal ganglion cells, and you can see in the healthy 0 hour, we have lots of expression, and then they subsequently lose that expression after days after damage. So we are doing the damage. So all of this to say that the damage response is equivalent in the species. So now we can really focus on specifically what happens in the spiny mice and specifically how their Mueller glia respond. So if you recall, in regenerating species such as the zebrafish, those Mueller glia respond to the injury in a really regenerative manner. But in mice, we do not see that. So the first thing that we wanted to look for, if you recall, those Mueller glia have to proliferate. So they have to divide to produce those daughter cells that can turn into those lost neurons. So really excitingly, when we look inside our retinas of the spiny mice, we see proliferating Mueller glia. So we see those Mueller glia that are undergoing that cell cycle. And you can see that here in these images. The green represent our Mueller glia cells. And we use a stain for proliferation, so a tag that'll label those proliferating cells in purple. And I hope that you can see we have lots of cells that are expressing both green and purple, indicating that we have Mueller glia that are undergoing cell division. And quantification here shows that we have lots of Mueller glia that are undergoing that cell division, and gene expression for multiple other cells associated with cell cycle genes are also upregulated in our Mueller glia after injury. So we have Mueller glia proliferation, so that first step required in zebrafish for regeneration is happening innately in our spiny mice. Additionally, not only are they upregulating genes for cell cycle, they are upregulating genes for regeneration as well. Genes like the proneural transcription factor, ASCL1, shown here in purple, required in zebrafish, absent in mice, and we see this really nice visual of it being upregulated in our Mueller glia after injury. The Mueller glia here, again, stained in green, and that proneural gene in the purple, and you can see them lining up really nicely. And again, quantification of that shows we have quite a few Mueller glia that are upregulating this proneural factor. Additionally, along with the proneural genes, our Mueller glia are also upregulating genes associated specifically with retinal cell regeneration and axon guidance, as well as just overall neurogenesis, so the birth of neurons. So this is really exciting. These Mueller glia seem to be responding in this regenerative way. But finally, to kind of wrap this all up nicely and coming back to where it started with the ganglion cells, spiny mice actually show a recovery in their retinal ganglion cells after injury. So you can see here again, our ganglion cells are lit up nicely with this green stain, and you see in our healthy control retina that layer of ganglion cells. And then what I hope you can appreciate is, as I showed you before, a few days after injury, we have that depletion of the retinal ganglion cells. And you can see over time, and really nicely highlighted at the 4-month mark post-injury, we see a recovery of these ganglion cells. Not only of the density of ganglion cells, but we also have a recovery of expression associated with mature ganglion cells. So I hope that this data excites you as much as it excites me, because it looks like for the first time we can show a mammal that has this pro-regenerative response after retinal injury, including this very regenerative response of the Mueller glia, where we have them proliferating, so dividing to create these daughter cells. But not only that, we have these genes upregulated that are associated with regeneration that we see in the zebrafish. And I really hope that I have shown you today that the spiny mice are a really promising new model for retinal regeneration and hopefully one day can help impact the treatments that we have for retinal degenerative diseases. Thank you.